Hello. Am I audible? The student who have joined, please let me know if I am audible. Yeah, thank you. Please, can you confirm that if the presentation is visible to you? Kindly let me know if you can see the presentation. Okay, okay. Thank you so much. We will wait till other students join and then we will start today's session. Hello, very good afternoon to the students who have joined. Please wait till it's 4 p.m. and then we will start today's session. Since I'm recording the session, I have muted your microphones and cameras also so that I can record it seamlessly. So if you have anything to say to me, you can let me know through the chat box. Okay, please wait. I will start the session at sharp 4 p.m. And let me know in the chat box if everything is fine with the voice and the PPT also. Please wait till it's 4 p.m. Then I will start the session. Meanwhile, if you have any doubt, any problem from previous day, you can let me know through the chat box also. Okay, so I think I will start with the introductions. Meanwhile, I have made the call open so that if other students want to join, they can join easily. Okay, so I think I will starting my introduction. So, very good afternoon to all. I am Mousumi Deb from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry, and today it is our week 2 live session. So, before starting the session, I want to clear out few points. Okay, so because every day we have new students, so that's why I have noted down here itself. So every week there will be a problem solving session, and these sessions will be on every Friday from 4 p.m. to 6 p.m. Okay, and these sessions are live on Google Meet platform. The link is this one which you have used for joining today's class. So this link will be same for our entire course or every Friday problem solving session, which is scheduled from 4 p.m. to 6 p.m. We'll use this same Google Meet link. Okay, so you can write it down anywhere. You can copy it any place. So that, okay, second. Yeah. So for every day, we will use the same Google Meet ID. So you can copy this ID, which I'm showing here on screen, and you can use it for every Friday for joining the class. So one two very important things are like that there can be network disruption here, okay, during our class. So if the network disruption happens from my side, okay, so please do not leave the session immediately. Please wait for 4 to 5 minutes. I will rejoin as soon as possible. And since I'm recording the session, I have muted your microphones and also the camera so that I can record the session seamlessly. But the chat box is always open, and I'm monitoring the chat box every now and then. So if you have any doubt, any query, anything, you can let me know through the chat box. Okay. I hope my voice is audible to you and also the presentation is visible. If not, you can let me know through the chat box itself. Okay. After the class, if you have any doubt or persist, okay, after class, you can let me know through the email. I will share my email ID at the end of the session so that you can use that email ID for and asking your queries. Otherwise, you can let me know your doubts in the class itself also. Okay, in the chat box, you can know. Let me know the what is your problem with a particular question or particular topic. I will try to answer your questions. Okay, this presentation and this class recording will be shared with you. Now, where you have to find this class recording and the presentation? Just you have to go to the medicinal chemistry. I hope my screen is visible to you. This is the medicinal chemistry where you find your announcement, course, and everything. So there is a left panel which shows the course outline. So here you will find after this week 2, then week 3, you will find there is a problem solving session tab. So if you click on there, you will directly redirected to a page. So if you click there, you will find this kind of place where for the Friday class you will get all the links and everything here. Okay, on this proportion. So you can download it from that particular file. You can download your problem solving session lecture video, and yeah, you can let me know your request here also before starting the session. Lectures, I'm afraid I have to take it in the English because I don't know if we have the provision to take the lectures in Hindi. But if you have any doubt or any query, you can ask in Hindi through the email. Okay, I will provide my email ID. So if you have any problem or any doubt, because there are many people from overall the India, so I cannot take the class in any regional language, right? I have to take it in English itself so that everybody understand. But if you have any problem, any doubt, you can let me know through the email. I would try to answer your queries through the emails also. So any problem? Uh, Weekly lectures means the one the professors is given, right? That is also, I don't think it is possible to do it in Hindi because it is already is recorded in English, so I don't think it will be available in Hindi. Okay. But if you have any doubt in understanding, you can let us know in the problem solving session. We will try to answer your query. Okay. So I think now doubts are clear. So we can directly go to the class. Okay. So I hope the presentation is visible, right? Any problem? If any problem is there with the presentation, let me know. Okay, so let me start the session. So as I have said, if there is any network issue from your side, you can rejoin it at any time because I'm recording the session. You will not miss anything. And if it is from my side, please wait for some time. Okay, I will rejoin as soon as possible. So today it is our week 2 live session. So again, I'm going to introduce myself. I think many people, many students have joined for the first time. So hello, my name is Mousumi Deb. I'm the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. And today in our week 2 live session, what we will do is we will discuss the topics which are related with the week 2 course content of this medicinal chemistry. But what are the course content of second week? There are enzyme catalysis, which is divided into part 1 and part 2. Then we have binding forces, protein structure and function. Then we will discuss the introduction to receptors, receptor types, their functions, receptor binding interaction, and ion channels. So how we do in this problem solving session is like that. I will show you around 20 questions, okay, one by one, and every question is a MCQ type question, and there will be 4 different answers for the each question, and only one with only one correct answer. After showing a particular question, I will give you some time. Maybe I will give you 3 to 4 minutes of time. You will read and think about read all the options very carefully, and you have to write what is the correct option for that particular or what is the correct answer for that particular question in the chat box. Okay, so A, B, C, or D, 4 options are there. You have to let me know through the chat box which of the following options are correct. And after checking your answers, what I will do is I will show you the correct answer, and we will discuss that particular question or that particular topic in detail, so that when in final examination you are asked some kind of subjective question from that same topic, you can answer it correctly. Okay, that is why but your weekly assignments are in MCQ, so we will practice the MCQs here. But the answers which I will show you, I will discuss it in the such a manner so that you can write it in your examination also. Okay, so let us start with the session. And yeah, one more thing, you have to submit your assignments on time, because there are marks on as submitting the assignment, but there will not be any kind of marks for answering into the during the live session. But I want all of you to answer the questions. Even if your answer is wrong, it does not matter, since there is no marking system. But answering is very important, otherwise I won't understand that if you are following the class or not. Okay, so let us start with the first question of today's session. So the first question of today's session is, which one of the following statement regarding enzyme is not true? Your options are: number A, enzymes are one kind of proteins. Number B, they can change the rate of the reaction. Number C, they get consumed in the course of the reaction. And number D, they are site specific in nature. So which of the following options are correct regarding, which of the following options are not true, or which of the following options are incorrect regarding enzyme? Again, I will repeat the options or the statements. Number first statement says that the enzymes are one kind of proteins. Number B, they can change the rate of the reaction. Number C, they get consumed in the course of the reaction. And number D, they are site specific in nature. So which of the following statements regarding the enzymes is not true? I am giving you some time. Read all the options very carefully, think, and then let me know in the chat box A, B, C, or D which of the following options or the statements are incorrect regarding the enzymes. In the chat box, let me know A, B, C, or D which of the following are correct option. Enzymes are one kind of protein, which is number A. Number B, they can change the rate of the reaction. Number C, they get consumed in the course of the reaction. Number D, they are site specific in nature. A, B, C, or D, which of the options or which of the statements is correct, is not correct regarding the enzymes? Okay, let me check the chat box. Okay, I have only got three answers in the chat box. Okay, so should I repeat the questions? So I get number C, then number C also. Okay, if you have any problem, you can let me know through the chat box. Okay, I have muted you all so that I can record the session without any problem. So I have got three, number C as the answer. Others? Anybody else want to answer this question? Okay, let me show you the answer and then let us discuss about the enzymes in detail. So the correct answer, the statement which is not true regarding the enzyme is, yes, it is number C, which says that they get consumed in the course of the reaction. So what is the enzyme? So basically, I think you have heard this term catalyst also. So what is the catalyst? Catalyst which catalyzes a reaction, which makes a reaction move faster. Okay, so enzyme is the similar one, but it catalyzes the biological transformations. Okay, so what is the enzyme? So enzyme is a protein which increases the rate of the reaction and which catalyzes a particular reaction by lowering the activation energy of a particular reaction. Okay, so what is now let us discuss this is called enzyme. Okay, so every enzyme is kind of a protein. Okay, so there are different kind of enzyme with a different size and shape and everything, but all the enzymes, they are basically a protein. For a particular enzyme, they have a particular active place which is, or a cleft which is known as the active site. So what is the active site of the enzyme? The active site of the enzyme is such a place where a particular substrate molecule binds and then the reaction happens and we get the product molecule. Okay, so this is the process of enzyme catalysis. The first step is the binding of the substrate to the enzyme active site, then the reaction where the substrates are converted to the product, and then third step is the release step where the product is released from the enzyme. So how this enzyme affects the particular activation energy of a reaction, you can say here from this figure, this graphs which demonstrates the stabilization of the reaction's transition state by a enzyme. Okay, so whenever there is a reaction, every starting material, they have to gain a particular energy which is known as the activation energy to reach to a state which is called the transition state, from where the products are formed. Okay, and what does this particular enzyme do? They actually lowers the activation energy of a particular reaction to reach a new transition state which has the lower energy, so the reaction gets reaction gets faster and we have the product formation with much ease. So enzymes provide a reaction surface and a suitable environment. They bring the reactants together, position them correctly so that they easily can attain their transition state configurations. Enzymes weaken the bonds in the reactants. They may participate in the reaction mechanism. Enzymes form stronger interaction with the transition state than the substrate or the product. Okay. So there are two theories. Okay, so whenever we are learning about the enzyme, let us understand about the two theories. The two theories about this enzyme substrate bindings are there. One is called as the lock and key theory and another is the induced phase hypothesis. So what is the lock and key theory? So the lock and key theory, it says that, okay, the lock and key theory says that the substrate and the enzyme active site, they are of the perfect fit. Okay, means just a lock and a key. When there is a lock, the lock has a perfect cleft where a particular key that insert and we can open the lock. So that is how the enzyme and substrate behave like that, and the enzyme active site, it fits perfectly with the substrate. But in the induced fit hypothesis, it says that the enzyme active site is not just like a lock and key, the exact match are not there, but whenever there is a substrate nearby to the enzyme, the active site will change or modify itself a little bit so that the substrate can accommodate to the active site and we get that particular product formation. So that is known as the induced fit hypothesis of the enzyme substrate binding. We will discuss this hypothesis in detail in later to our today's class. Okay, so this is all about this enzymes and enzyme binding. I have told it earlier also. If you have any doubt or any query regarding any question, let me know in the chat box. Okay, so I can answer your queries then and there. Otherwise, at the end of the session also, you can ask me your queries all together. Okay. So for the first question, number C was the correct answer. Now I will go to the next question. So problem number 2, it says that what is the correct graph for the enzyme control reaction versus substrate concentration? Your options are this: number A, number B, number C, or number D. There are, on the y-axis we have the rate of the reaction, on the x-axis we have the substrate concentration. Now there are 4 graphs: number A, number B, number C, and number D. You have to let me know that which of the following graph is the correct for a enzyme catalyst reaction. In the previous question, I have got only 3 answers in the chat box. In this question, I want all of you to participate and answer, even if your answer is wrong. Okay, it is fine if your answer is wrong, since I am not giving you any marks regarding the answers, but I want all of you to participate and let me know which of the following is the correct graph for this enzyme control reaction rate and the substrate concentration. Again, I am giving you some time. In the chat box, let me know A, B, C, or D, which of the following is the correct answer. Which of the following is the correct graph for the enzymes control reaction versus substrate concentration? Okay, let me check the chat box now. Okay, I have got 3 different answers. Number A, number B, and number C. 3 different options I have got as the answer for this problem number 2. Okay, let me check which of the following is the correct one. So the graph which depicts correctly the enzyme catalyzed reaction rate and the substrate concentration is, it is the number A, the first graph. Okay, this is the graph for the enzyme's catalyzed reaction rate and the substrate concentration. So this is also known as the Michaelis-Menten enzyme catalyzed reaction graph. Okay, and this is the graph for this particular famous Michaelis-Menten enzyme catalysis. So this graph, it depicts many things. Okay, let us start one by one. So first one is in the y-axis we have the velocity or rate of the reaction. In the x-axis we have the substrate concentration, and this is the graph. So in the first phase of the graph, as you can see this position. So when the particular it depicts the substrate concentration is low. So what it means, when the substrate concentration is low, means there are lots of enzyme which are available. Okay, means this position, when the substrate concentration is low, the enzyme catalytic reaction, it follows the first order kinetics. Okay, then it follows the mixed order kinetics, and at the end, when all the enzymes are occupied, the enzyme or there is no enzyme to which are there are no enzymes which are left with their active site, means the reaction enzymes are saturated. There it follows the zero order kinetics. Why zero order kinetics? Zero order kinetics because now the reaction rate has reached to this maximum value, which is the Vmax, and by changing or increasing the substrate concentration, there is no change in the reaction rate. Initially, when the substrate concentration is low, as you increase the amount of the substrate, the velocity of the rate of the reaction will increase, and it will follow the first order kinetics. But at the end, when there is when the substrate concentration is high, in the particular reaction attains its highest velocity, which is the Vmax, and we do not get any kind of increase of the rate. That means it follows the zero order kinetics. Okay. The highest rate of the enzyme catalyzed reaction is known as the Vmax, and the half of that Vmax, okay, means the half of the highest rate, it's known as the Vmax by 2. The substrate concentration which is at the Vmax by 2 is known as the Km, which is known as the Michaelis-Menten constant. Okay. all these equations are not, the derivations are not in your syllabus, but the equations are very important, which you need to remember, at least the Michaelis-Menten equation, which I will discuss soon. Okay, so answer number A was the correct graph for the enzyme catalytic reaction versus substrate concentration. Okay, now let me go to the next problem. So problem number 3 says, which of the following statements is not true for Km and Vmax? Okay, Km I have already mentioned, it is the Michaelis-Menten constant, means Km is the substrate concentration here, Michaelis-Menten constant, Vmax is the highest velocity. Okay, Vmax is the highest velocity. So now let us see the statements. So statement number A says Vmax is the maximum rate at which a particular enzyme catalyzed reaction can proceed. Number B, a small value of Km tells us that the enzyme binds strongly to its substrate. Number C, Km is the concentration of the substrate at which the rate of the reaction reaches Vmax. And number D, a large value of Km tells us that an enzyme shows little specificity for a given substrate. You have to identify which of the following statements is not true. Means which of the following statement is false. Again, I will repeat the statements. Number A, it says that Vmax is the maximum rate at which a particular enzyme catalyzed reaction can proceed. Number B, a small value of Km tells us that the enzyme binds strongly to its substrate. Number C, Km is the concentration of the substrate at which rate of the reaction reaches to Vmax. And number D, a large value of Km tells us that an enzyme shows little specificity for a given substrate. Read all the options carefully. I will give you 2 to 3 minutes. Then let me know in the chat box A, B, C or D which of the following statement is not true regarding Km and Vmax. Let me know in the chat box A, B, C or D which of the following statement is incorrect. Which of the following statements is not true for Km and Vmax? Okay, let me check the chat box now. Okay, I think I have got a few answers for this question. Number C, number C, number D, number D. So I have two different options as answer, number C and number D. So let me check which of the following is the correct answer. So the statement which is incorrect regarding the Km and Vmax is number C, which says that Km is the concentration of the substrate at which the rate of the reaction reaches Vmax. But it is not that. In the previous slide, I have shown you that Km is the substrate concentration when the rate of the reaction reaches Vmax by 2, means the half maximum. Okay, so when the rate of the reaction reaches to half of this highest value, it is that the substrate concentration of that particular point is known as the Km or the Michaelis-Menten equation. Other statements, these are correct, means Vmax is the maximum rate at which the particular enzyme catalyzed reaction can proceed. A small value of Km tells us the enzyme binds too strongly to its substrate concentration, and a large value of Km tells us that the enzyme shows little specificity regarding a given substrate. So this is the Michaelis-Menten equation or and this is the Michaelis-Menten curve. Okay, so this equation is very important. This derivation of this equation is not so much important because it is not in your course in detail. But this particular equation is very important. So what is the Michaelis-Menten equation? This V0 equal to Vmax into the concentration of substrate divided by Km plus concentration of substrate, where V0 is the initial velocity in mole per time. S is the substrate concentration which is in molar, Vmax is the maximum velocity, and Km is the substrate concentration at the half Vmax. Okay, so this is the Michaelis-Menten equation, and this graph is known as the Michaelis-Menten curve. Okay, in here in the initial stage when the particular reaction when the substrate concentration is very low, so when the substrate concentration is much less than Km, we can neglect the Km in the denominator, means the equation will behave like this: V0 equal to Vmax into S divided by Km. Now, which means that the V0 changes with the it is a first order reaction. Okay, here. And then when at the end of the reaction, means when it achieves the Vmax, when the V0 equal to Vmax, so it is a zero order reaction, because then when the substrate concentration is much higher than the Km, we can neglect the Km here, and then this concentration is cancelled out, and we get this equation. Okay, so this equation is very important, and this curve is also very important, which you need to remember and understand also. So if you have any doubt or any problem regarding this equation and in this curve, you can let me know. Okay, you can let me know now, or at the end of the session also you can let me know if you have any doubt or any problem regarding this Michaelis-Menten equation and the Michaelis-Menten curve. Okay, so now I will go to the next problem. So problem number 4, it says that it is a mathematical question, okay, which says that what is the ratio of V0 Vmax by V0 when the substrate concentration equal to 9 km. Your options are number A 9 by 10, your option number B 10 by 9, option C 9 by 8, and option D 1 by 9. The equation, the Michaelis-Menten equation, you have to use here. If you use that equation, you can do it very easily. I will give you some time. Take a pen and paper, do it quickly, and let me know what will be the correct answer. What is the ratio of Vmax by V0 when the substrate concentration equal to 9 km? Option A is the 9 by 10, option B is the 10 by 9, option C is the 9 by 8, option D is the 1 by 9. The Michaelis-Menten equation, which I have shown you just now, you have to use that equation to solve this question. Try to solve this question. I will give you 2 to 3 minutes. Remember the Michaelis-Menten equation, which says V0 equal to Vmax into substrate concentration divided by Km by Vmax. Km by plus substrate concentration. You have to use that particular equation to solve this question. Try it. I will give you some time. Okay, let me check the chat box now. Okay, I have got few answers. Number B, number A, number B. So I have got one number A and several number B. So let me check if which of the following is the correct answer. So the correct answer will be S number B, 10 by 9. So this is a very easy question. Okay. So this is the Michaelis-Menten equation. I am again telling you, you have to remember this equation and you have to memorize it. Okay. And what is the equation? What are the terms are indicating? You have to know all. So V0 equal to Vmax into S divided by Km plus S. So what is the V0 is initial velocity, S is the substrate concentration in molar, Vmax maximum velocity, and Km is the substrate concentration at the half Vmax, which is also known as the Michaelis-Menten constant. So now if you just take the Vmax here in the left hand side, so we will get V0 by Vmax equal to such concentration of S divided by Km plus S. Since in the question says that S equal to 9 km, so you have to just replace the S with the 9 km. So we will get 9 km divided by km plus 9 km means 9 km divided by 10 km, km cancel out, and we have the V0 by Vmax equal to 9 by 10. So the Vmax divided by V0 will be 10 by 9. Okay, so number B was the correct answer. This type of easy mathematical questions will be asked in your examination and also in your assignment. So you have to practice this kind of questions. Okay. Now I will go to the next problem. So problem number 5 says, which coenzyme is required by the enzyme lactate dehydrogenase? Your options are number A NADP, number B NADH, number C NAD plus, and number D FAD plus. Which coenzyme is required by the enzyme lactate dehydrogenase? Number A NADP, number B NADH, number C NAD plus, and number D FAD plus. Which coenzyme is required by the enzyme lactate dehydrogenase? Everybody try to answer these questions. Again, I am saying even if your answer is wrong, it does not matter, but answering is very important. So A, B, C or D, which of the following is the correct coenzyme for the enzyme lactate dehydrogenase? NAD, NADP, NADH, FNAD plus or FAD plus? Which coenzyme is required by the enzyme lactate dehydrogenase? Okay, let me check the chat box now. Number B, number C, number B, number C, C, B, C. So I have mostly got two as answers. Okay, one is number B and then number C. So there are two options mainly as an answer. Let me check which of the following will be the correct one. So the correct answer is the coenzyme which is required by the enzyme lactate dehydrogenase will be It is number C, NAD+. So NAD+ is the coenzyme which is required by the enzyme lactate dehydrogenase. So, what is cofactors, coenzymes, and all these things? Let us discuss in detail. Okay, but first let us see what does this enzyme lactate dehydrogenase do. So, lactate dehydrogenase it converts the lactic acid to the pyruvic acid. Okay, so basically dehydrogenase it means that it is eliminating the particular hydrogen from the particular reaction. Okay, so basically it is catalyzing, it is doing the oxidation. So as you can see in the lactic acid we have this particular hydroxyl group here which is converted to the ketone group by the lactate dehydrogenase. Okay, so what is the coenzyme is required here? It is the NAD+. So this is the structure of the NAD+. Okay, and that is how this particular reaction catalyzes happens. Okay, this the NAD plus it gets reduced to NADH, and lactic acid it gets oxidized to the pyruvic acid. So this is the reaction which is catalyzed by the lactate dehydrogenase. So many enzymes they require an additional non-protein substance which is known as the cofactors for the reaction to take place. Deficiency of the cofactors can arise from the poor diet resulting in the loss of enzyme activity and subsequent disease like scurvy. Cofactors are either metal ions like zinc or small organic molecules or called the coenzyme like NAD plus or pyridoxal phosphate. Most coenzymes are bound to the ionic bonds and non-covalent bonding interactions, but some are bound covalently and are called the prosthetic groups. Coenzymes are derived from the water-soluble vitamins and act as a body's chemical reactants. For example, lactate dehydrogenase requires the coenzyme nicotinamide adenine dinucleotide, which is NAD plus, in order to catalyze the dehydrogenation of the lactic acid to pyruvic acid. NAD plus is bound to the active site along with the lactic acid acts as an oxidizing agent. During the reaction, it is converted to the reduced from NADH. Conversely, NADH can bind to the enzyme and act as a reducing agent when the enzyme catalyzes the reverse reaction. NAD plus and NADPH are the phosphorylated analogues of the NAD plus and NADH respectively and carry out the redox reaction by the same mechanism. NADPH is used in almost exclusively as the reductive biosynthesis, whereas NADH is used primarily for the generation of the ATP. Okay. So what is the difference between the coenzymes and cofactors? So coenzymes are the small organic non-protein molecules that carry chemical groups between the enzymes, and cofactor are the non-protein chemical compound that tightly and loosely binds with the enzymes or other protein molecules. Coenzymes are the type of cofactor. Coenzymes are prosthetic groups are two types of the cofactor. So cofactor is the it is in the broader sense. Okay, coenzymes is one type of cofactor, and also prosthetic groups are there which are also one type of cofactor. So coenzymes these are the molecules, and cofactors are the chemical compound. Coenzymes are mainly the organic molecule and which are loosely bound to the enzyme, and cofactors are the inorganic compounds which can covalently bound with an enzyme. Coenzyme assists the biological transformation. Cofactors aid the function of the relative enzyme. Coenzymes serve as the carriers to the enzymes and increase the rate of the reaction catalyzed by the enzyme. And coenzymes can be removed easily from the enzyme easily. Cofactor can be only removed by the denaturing of the enzyme. For example, vitamin, biotin, coenzyme A are the example of coenzyme. Are metal ions like zinc plus 2, potassium plus 2, magnesium plus 2 are the example of the cofactors. Okay. So this is all about the enzymes and cofactor. Now I will go to the next question. The problem number 6 again is a mathematical question. So it says that consider the following values of the Michaelis-Menten enzyme kinetics and calculate the dissociation constant for enzyme substrate complex and the catalytic efficiency of the enzyme. K1 equal to 4 into 10 to the power 8 mole per minute and inverse. K minus 1 into 10 to the power 3 minute inverse. K2 equal to 6 into 10 to the power 3 minute inverse. And these A, B, C and D are the options for your dissociation constant for the enzyme substrate complex and the catalytic efficiency. So A equal to 0.5 micromolar and 8.57 into 10 to the power 8 mole per minute inverse. B equal to 2.5 micromolar and 8.57 10 to the power 8 minute per mole inverse. C equal to 2.5 micromolar and 0.857 into 10 to the power 8 minute per mole in molar per minute inverse, and 5 into 5 micromolar and 0.857 into 10 to the power 8 molar per minute inverse. So you have to identify the correct answer. Now let me know. Let me just show you what is the particular thing is this K1, K inverse, and K2. So we have enzyme, right? We have free enzyme. Okay, let's just 1 second. And let me. So just let me for I think most of you know, but the one who don't know. So let me just show you. So any enzyme catalyzed reaction. So it has a basic reaction which is enzyme, and we have the substrate, right? So enzyme and substrate, in the first step, it forms a enzyme substrate complex. It is an active species, this enzyme substrate complex. This particular enzyme substrate complex formation is a reversible reaction. So the forward rate constant is the K1, and the backward rate constant for this step is the K in -1. And then the second step, which is the formation of the product P, and we have the enzyme without any change. The rate constant of this particular step is the K2. All this K1, K inverse and K2 values are given here. Now you have to calculate what is the dissociation constant for the enzyme substrate complex and the catalytic efficiency of the enzyme. That you have to find out. The values are given. I have shown you what is K1, K -1 and K2. Now you have to find out what is the values for the dissociation constant and the catalytic efficiency. I will give you some time for this reaction, for this question. Try for some time. Let me check the chat box. Okay. Okay, I think this question might be a little tough for you guys. So it's fine. I will show you how to solve this kind of question so that in future, if this type of question is given, you can solve it easily. Okay. 1 second. Okay, so the correct answer was number C, which says 2.5 micromolar and 0.857 into 10 to the power 8 molar minute inverse. So you have to remember these two things. Okay, one, the dissociation constant for an enzyme substrate complex is given by K minus 1 divided by K1. Okay, just you have to take K minus 1 and you have to divide it with K1 and you will get this this particular thing, which is, just let me highlight this thing. So the dissociation constant is given by K minus 1 divided by K1. Okay. So when the values of the K1, K minus 1 all are given, you can easily get the value, which is this one. And the catalytic efficiency of a particular enzyme is given by this K2 by Km. Okay. Now K2 is given here. So what is the Km? Km is the Michaelis-Menten constant, which is given by this K minus 1 plus K2 divided by K1. So this is the Km. So first you have to calculate what is the Km from these given values, and then you have to calculate the catalytic efficiency. Okay, which is this one. So these things you have to know, or you have to remember that what is the catalytic efficiency, what is the dissociation constant, and what is the Km, all these things. Okay, so the basic constants are this K1, K minus 1 and K2, and all the values you can get if you know the equations and if you have the values of these things, or vice versa. Means the question can be given like you are given the catalytic efficiency and then you may be asked to calculate the K2, or if Km is given, you have to or K2 is given and catalytic efficiency is given, you may be asked to calculate the Michaelis-Menten constant. This thing also can be come in your examinations. Okay, so just remember these two things. Now next question, in which family of enzymes does fumarase enzyme belong? Your options are number A ligase, number B lyase, number C isomerase, and number D transferase. In which family of enzymes does the enzyme fumarase belong? Ligase, lyase, isomerase, transferase. Let me know in the chat box which of the following options are correct. In which family of enzyme does fumarase enzyme belong? Ligase, lyase, isomerase, or transferase? In which family of enzymes does fumarase enzyme belong? Okay, let me check the chat box. Okay, I have got several answers. Explain 6 question. Problem number 6. Okay, the previous question, right? Okay, I will re-explain it. Don't worry. Let me check the first answers of this question, then I will re-explain the problem number 6. So number B, number C. So for the problem number 7, I have got the answers like number B and number C. Okay, I am checking that later. So let me just explain this particular thing. So in an enzyme catalyzed reaction, this is the basic reaction. Okay, this reaction which I have written here, this is a basic reaction, E plus S, which is enzyme plus substrate. Then the forward it forms the enzyme substrate complex. This is a reversible reaction, means the enzyme substrate complex, it is in the equilibrium with this particular enzyme, free enzyme and substrate. Okay, the forward rate constant for this reaction is the K1 and the backward rate constant is the K minus 1. Okay, and then the forward rate constant for the product formation is the K2. All the values of K1, K minus 1 and K2 are given here. You are asked to calculate the dissociation constant. You were asked to calculate the dissociation constant for the enzyme substrate complex and the catalytic efficiency of the enzyme, right? So these are the formulas for the dissociation constant and the catalytic efficiency. So the dissociation constant of the enzyme substrate complex, which is the ES, this one is the enzyme substrate complex. The dissociation constant for this enzyme substrate complex is K1 divided by K minus 1 divided by K1. So what is the value of K minus 1? 1 into 10 to the power 3 minute inverse, right? This is the value of the K minus 1. And for the K1, the value is 4 into 10 to the power 8 mole per minute inverse. Okay, so then if you just do it the reversible, okay, just you have to just divide it by it and you will get this value, which is 2.5 into 10 to the power minus 5 molar or 2.5 micromolar. Now the catalytic efficiency, the constant for the catalytic efficiency, we have the value. The particular formula for calculating catalytic efficiency is the K2 by Km. Okay. Now this Km is the similar. The Km we have already discussed many times. This Km is the Michaelis-Menten constant. Okay. So Km, I am showing you the Km equal to the substrate concentration at the half Vmax. But what is the value of the Km? So the value of the Km like this. The value of Km is K minus 1 plus K2 divided by K1. Okay. So if you just K minus 1 is the 10 to the 1 into 10 to the power 3 minute inverse, K2 is the 6 into 10 to the power 3 minute inverse. So what is the value, and divided by K1, which is 4 into 10 to the power 8 mole per minute inverse. Okay, so what is the value of this particular thing? So what the value goes like this. This is the 7 into 10 to the power minus 5. Okay. One second, I have to check one thing. Okay, I think there is some error in calculation. Just let me recheck the thing once. Okay, there is a typographical error here which I have just missed. Okay, so Km equal to K minus 1 plus K2 by Km. Okay, this is a 1 plus 6 into 10 to the power 3 divided by, so it will be not 7, it will be just 1 second. It will be 7 by 4 into 10 to the power minus 5. 7 by 4 means it will be 1.75. Okay, it will be 1.75. And here also the catalytic efficiency will be K2, which is 6 into 10 to the power 3. K2 divided by, here it will be 1.75. Sorry for the mistake. There was a typographical error. Thanks for pointing out. I think that I just missed this thing. So it will be 6 divided by 1.75, which means 3.42. Okay, it will be 3.42. Okay, I think I had done this particular mistake, so I have just corrected it. So you can recheck it by yourself. Okay, if I am doing it right or not. So yeah, I think I have made this mistake. So as you can see here, Km equal to this formula. 1 second, just let me use the pen here. So Km equal to your K minus 1 plus K2 divided by K1, right? So this K minus 1 equal is 1 into 10 to the power 3, and then K2 is 6 into 10 to the power 3, means we will get 7 into 10 to the power 3 divided by 4, which is K1 equal to 4 into 10 to the power 8. Okay, 4 into 10 to the power 8. So we will get the value of 1.75 into 10 to the power minus 5. Okay, this is the value of the Km from this equation, and for the catalytic efficiency we have to just divide the K2 divided by Km. So this is the value of K2, and if you divide this K2 by Km, so you will get this value. Okay, so it was my mistake. I have just done an error for calculation error. So I think now it is fine. Okay, one second. Just let me know. So now is it clear? Can you let me know if now the problem 6 is clear to you? I had done a little calculation error, so I'm very sorry for that. So now I think the problem number 6 is clear. Whoever have asked to re-explain it, can you just confirm if it is now fine? Okay, I think it is now fine. When I will share this PPT with you, you can check it, okay? You can check it by yourself. Thank you for pointing out the mistake. It was a calculation error. So when I will submit the PPT, or you can check from here or there also, I think it is very easy. Just you have to put the values correctly to the formula, okay? So now let us go to the problem number 7, which I have to explain now. So yeah, I have asked you about in which family of enzymes does the fumarase belong. So you have written that number B and number C. There were answers that number B and number C. So now let me check which is the correct answer. So the family where the fumarase enzyme belong is, it is number B, lyase. Lyase is the family of the enzyme where the fumarase enzyme belong. So what is the role of the fumarase enzyme? So basically the fumarase, what does it do? It converts the fumarate, which is this one, to the S-malate. Okay, the fumarate to S-malate, it is the reaction. So there are different class of the enzyme. So you have to remember which class of the enzyme perform which type of reaction. What is the example? So first class is the oxidoreductase. So oxidoreductase, as their name suggests, they do the reduction, oxidation or redox kind of things, like lactate dehydrogenase, which I have discussed just in the last question, I think. So these are called the oxidoreductase, which catalyze the oxidation reduction reaction. Then we have transferase, which move the chemical group like hexokinase. The hydrolysis, which catalyzes the hydrolysis, the bond cleavage with the transfer of the functional group of water, for example lysozyme. Then we have the lyases. They are the non-hydrolytic bond cleavage, for example fumarase. Then we have isomerase, which perform the intramolecular group transfer or isomerization, like triose phosphate isomerase. And then we have ligase, which does the synthesis of the new covalent bond between the substrate using the ATP hydrolysis, like RNA polymerase. Okay, so these are the classes of different enzymes, their reaction type and their examples. So fumarase, it falls in the enzyme class of lyase. What does it do? Non-hydrolytic bond cleavage is performed by the fumarase. Okay. So lyase and ligase, these are very important class of the enzymes, but most of the students I have seen that they get confused between these two enzyme class, the lyase and ligase. Sometimes they just confuse that which one is lyase and which one is ligase because their names are so similar. So basically lyases are the enzyme that catalyze the bond breaking reaction. Okay, and ligases are the class of the reaction which is involved in catalyze the bond formation. So this is a very crucial difference between lyases and ligases. Lyases breaks the enzyme that catalyzes the bond breaking, and ligases catalyzes the bond making. Lyases cause the forming of the double bond. Ligases cause the forming of the chemical bond. Lyases involved in the elimination reaction. Ligases are involved in the hydrolysis reaction. Lyases act on a single substrate per reaction. Ligases act on a two substrate or reactant per reaction. And lyases are useful in glycolysis, and ligases are important in the DNA replication. Okay, so these are the differences between the lyases and ligases. So these enzyme class, different class of the enzyme, their example and what kind of reaction they perform are very important, which you have to remember. Okay. So next I will go to the next problem. Problem number 8 says, in the TCA cycle, the conversion of the malate to oxaloacetate is catalyzed by dash. You have to fill the blank. Okay. Your options are number A, malate dehydrogenase, number B, malate dehalogenase, number C, oxaloacetate synthesis, and number D, aconitase. Which of the following enzyme catalyzes the conversion of malate to oxaloacetate in the TCA cycle? Your options are number A, malate dehydrogenase, number B malate dehalogenase, number C oxaloacetate synthesis, and number D aconitase. Let me know in the chat box A, B, C or D which of the following is the correct option. In the TCA cycle, conversion of malate to oxaloacetate is catalyzed by which enzyme? Malate dehydrogenase, malate dehalogenase, oxaloacetate synthase, aconitase. This PPT and the class recording, which I am it will be available. It will be made available to you in the SWAM portal. At the end of the session, I will show you. Okay, and at the end of the session, I will also share my email ID so that if you have any doubt or any query regarding today's session or the course content, you can let me know through the email also. Let me now check the chat box. Okay, I think I have got few answers. Number C, number C, number A, number C. So I have got two number C and one number A and three number C and one number A as the answer. Okay, let me check which of the following will be the correct answer. So the enzyme which catalyzes the conversion of malate to oxaloacetate in the TCA cycle is, it is number A, malate dehydrogenase. So, malate dehydrogenase is the enzyme which catalyzes the conversion of L-malate to oxaloacetate. So, this is the structure of malate. Okay, and malate dehydrogenase, again dehydrogenase is which kind of enzyme it is? It is an oxidoreductase class of family which catalyzes the oxidation reaction. It oxidizes the L-malate to oxaloacetate. And what is the coenzyme? Just similar as the lactate dehydrogenase, here also the coenzyme is the NAD plus which is converted to NADH in the course of the reaction. Okay. So now this is the TCA cycle. Okay, and the one so is first is the pyruvate conversion of pyruvate to acetyl CoA by pyruvate dehydrogenase. Okay, it is also a dehydrogenase, means it is also a an oxidation reaction type of reaction. Here also NAD plus is the coenzyme. Then followed by the conversion of acetyl CoA. Now acetyl CoA acts as a acts here, and it takes part in the conversion of oxaloacetate to citrate. So first the citrate is converted to isocitrate by the isocitrate dehydrogenase. Okay, and then the citrate is converted to isocitrate by the aconitase. Isocitrate conversion to oxalosuccinate by the isocitrate dehydrogenase. Then oxalosuccinate to alpha ketoglutarate. This is the catalyzed by the isocitrate dehydrogenase. Then alpha ketoglutarate to succinyl CoA. It is catalyzed by the alpha ketoglutarate dehydrogenase. Succinyl CoA to succinate. It is catalyzed by the succinyl CoA synthetase. Then succinate to fumarate converted by succinate dehydrogenase. Fumarate to malate, it is converted by the fumarase. Malate to oxaloacetate is converted by malate dehydrogenase. And then oxaloacetate to citrate, it is converted by citrate synthetase. Okay, and here acetyl CoA takes part, which is converted from the pyruvate. So this is the TCA cycle, and there are different enzymes are involved in different conversions. Okay. Now let me go to the next slide. So problem number 9 says the enzyme cyclase is activated by dash. You have to fill in the blanks. Your options are number A ATP, number B H2O, number C NO2, number D NO. The enzyme cyclase is activated by what? ATP, H2O, NO2 or NO? The enzyme cyclase is activated by which of the following things? ATP, H2O, NO2 or NO? Let me know in the chat box A, B, C or D which of the following you think is the correct answer. Enzyme cyclase is activated by which of the following? ATP, H2O, NO2 or NO? Okay, let me check the chat box now. Okay, I have got few answers. Okay, number A and number D, one number D and others are number A, which I have found. Okay, let me check which of the following is the correct. Okay, another one number D. So I have got two number D and other I have got number A. Okay, let me now check which of the following will be the correct answer. So for problem number 9, the correct answer is it is number D, NO. So enzyme cyclase is activated by NO. So I have got one number D and others were number A, so it's fine. So the external control of enzymes is usually initiated by the external chemical messengers which do not enter the cell. However, there is an exception to this. It has been discovered that the cells can generate the gas nitric oxide by the reaction sequence shown in figure 1, this one, and which is catalyzed by the enzyme nitric oxide synthetase. Okay, because nitric oxide is a gas, it can diffuse easily through the cell membranes into the target cell and activate enzymes called cyclases to generate the cyclic GMP from GTP. Cyclic GMP then acts as a secondary messenger to influence other reactions within the cell. By this process, nitric oxide has an influence on the diverse range of the physiological processes, including the blood pressure, neurotransmission, and immunological defense mechanism. Okay, so this is from arginine to citrulline is formed, and here from by this reaction the nitric acid is nitric oxide are synthesized, and this nitric oxide it activates the enzyme cyclase. Okay, and the cyclase it converts the guanosine triphosphate or the GTP to the cyclic GMP. Okay, and this is also known as the external control of the enzymes. So nitric oxide has a very crucial role in the cellular biology, which is involved in the different methods. Like it inhibits the platelet aggregation, it protects the brain tissues via the hemodynamic regulation, induces the vasodilation and increases the blood flow. It also is a mediator of the migraine and other vascular headaches. It protects the mucosal lining of the gastrointestinal system and mediates the macrophage cytotoxicity against the microbes. Okay, so the correct answer was the number D, NO. So I think I have got few number D or few NOs. Okay, so it's fine. Why ATP cannot use? So it's not about why, because ATP is not used. Okay, ATP is not it cannot activate. It is not about why. Okay, because it is like it's like it does not do it. ATP does not perform this particular reaction. It is not about why. Okay, so I think it is not clear. It's not why. It is about more about the what, because ATP does not do this reaction. ATP do not activate this. Only nitric oxide can activate this particular enzyme cyclase. Okay. Yeah. So some things like this, like you can ask that why ATP cannot. So these things are not like the why. So not that every reaction will be activated by ATP or nitric oxide. So this is like the specific reactions are done by specific enzymes, and specific enzymes are activated by the specific reagents. Okay, so for that particular enzyme, NO is the activator. Okay. So now I will go to the next problem. So problem number 9 says, what is the catalytic triad of the amino acids in the active site of the enzyme chymotrypsin? Your options are number A, serine, arginine and glutamine. Number B, threonine, histidine and methionine. Number C, serine, histidine and aspartic acid. And number D, threonine, arginine and glutamic acid. What is the catalytic triad of amino acids present in the active site of the enzyme chymotrypsin? Number A, serine, arginine and glutamine. Number B is a threonine, histidine, methionine. Number C, serine, histidine, and aspartic acid. And number D is a threonine, arginine, and glutaric acid. In the chat box, let me know A, B, C, or D, which of the following is the correct answer. So, what is the catalytic triad of amino acids present in the active site of the enzyme chymotrypsin? A, B, C, or D, let me know in the chat box which of the following will be the correct answer. Okay, let me check the chat box. So yeah, these questions are not only important for the exam point of view, it is also important for your assignment point of view and for the understanding of this course better, okay? So let me check the answers. I have got number C. Most of the answers are saying it is the number C. And also I have got two number A. Okay. Let me now check which of the following will be the correct answer. I have got number A and number C as the answer. So the correct answer, the catalytic triad of amino acid which is present in the active site of the enzyme chymotrypsin is, it is number C, serine, histidine and aspartic acid or aspartate residue. So amino acid, serine, histidine, okay, 1 second. Yeah. So this is the hydrolysis of the peptide bonds catalyzed by the enzyme chymotrypsin. So chymotrypsin is the enzyme which is involved in the hydrolysis of the peptide bond. So the catalytic triad are present is a serine, histidine and aspartic acid. So first there is a serine which has a hydroxyl group here which involves in the nucleophilic attack on the carbonyl carbon of a peptide bond. Then it follows it forms a tetrahedral intermediate and then the histidine. So now this aspartate residue here, it actually, what does it do? It actually pulls the proton from the histidine residue so that this nitrogen lone pair gets more basic and it attacks this hydrogen from this serine residue so that we can have this kind of intermediate form. And then what happens, this intermediate breaks and the hydrolysis of this peptide bond happens and the regeneration of the catalysis happens by using a catalytic water which is present in the active site. Okay, so the amino acids serine and cysteine are present in the active site of some enzyme. These amino acids have nucleophilic residues like OH and SH respectively, which are able to participate in the reaction mechanism. They do this by reacting with the substrate to form an intermediate that would not be formed in the uncatalyzed reaction. These intermediates offer an alternative reaction pathway that may avoid the high energy transition state, hence increase the rate of the reaction. Normally, an alcoholic OH such as the one in the serine is not a good nucleophile. However, this is usually a histidine residue close by to catalyze the reaction. For example, the mechanism by which the chymotrypsin hydrolyzes the peptide bond involves the catalytic triad of the amino acids serine, histidine and aspartic acid. Serine and histidine participate in the mechanism as nucleophilic and acid-base catalysts respectively. The aspartate group interacts with the histidine ring and serves to activate and orient it correctly to the mechanism. The presence of the nucleophilic serine residue means the water is not present, it is not required in the initial stages of the mechanism. This is important firstly because water is poor nucleophile and may also find it difficult to penetrate in the occupied active site. Secondly, a water molecule would have drifted into the active site, search out of the carbonyl group before it could attack it. This was something similar to the game of the blindsman bluff. The enzyme, however, can provide a serine OH with the position exactly the right spot to react with the substrate. Therefore, the nucleophile no need to search for its substrate. The substrate can be delivered to it. Water eventually is required to hydrolyze the acyl group attached with the serine residue. However, this is a much easier step than the hydrolysis of the peptide bond, as esters are more reactive than amides. Furthermore, the hydrolysis of the peptide link means one half of the peptide can drift away from the active site and leave room for the water molecule to enter. A similar enzymatic mechanism is involved in the action of the enzyme acetylcholinesterase and pancreatic lipase and a viral protease enzyme carried out by hepatitis C virus. Okay. So this is the mechanism of action which is followed in the active site in the enzyme chymotrypsin by the triad amino acids serine, histidine and aspartate acid. Now I will go to the next problem. So problem number 11 says what kind of inhibitor glucose 1-phosphate is for the enzyme phosphorylase A? Your options are number A reversible, number B allosteric, number C suicide and number D irreversible. What kind of inhibitor glucose 1-phosphate is for the enzyme phosphorylase A? Your options are like this: number A reversible, number B allosteric, number C suicide and number D irreversible. What kind of inhibitor glucose 1-phosphate is for the enzyme phosphorylase A? Reversible, allosteric, suicide or irreversible? This enzyme inhibition and all this thing we will discuss in detail in the next week. It's in the week 3 live session. But today I thought, so let's see if you are have learned anything about the enzyme inhibition till now. Okay, let me check the chat box. Okay, so I have got few answers, number D and number B. Two different options I have got as answer. Let me check number B or number D, which of the following is the correct one. So, the inhibitor glucose 1-phosphate for the enzyme phosphorylase A is kind of number B, allosteric inhibitor. So basically, allosteric inhibitor or allosteric site of a particular enzyme is the allosteric site is a site which is not the active site of the enzyme. So active site of an enzyme where the substrate binds, we know all. Okay, but allosteric site is a site which is different, and some inhibitor molecules, they bind to so inhibitor molecule actually they can bind to the active site or they cannot bind to the active site. So when an inhibitor does not bind to the active site but bind to another site and changes the active site make the active site shut, or changes the conformation of active site, is known as the allosteric inhibitor. So for example, the phosphorylase A enzyme, its catalytic activity is controlled by the internal control of the glucose 1-phosphate or AMP, and it is a allosteric inhibitor. Okay, so virtually all enzymes are controlled by agents which can either enhance or inhibit their catalytic activity. Such control reflects the local condition within the cell. For example, the enzyme phosphorylase A catalyzes the breakdown of the glycogen, the polymer of the glucose monomers, to the glucose 1-phosphate subunit. It is stimulated by the adenosine 5 monophosphate or AMP and inhibited by the glucose 1-phosphate, thus raising the level of the product. Glucose 1-phosphate acts itself as a self-regulating or brake on the enzyme. So how does this control takes place? The answer is this: many questions have a binding site which is separate from the active site and known as the allosteric binding site. Now that is where the agents controlling the activity of the enzyme bind. When this occurs, an induced fit takes place which alters not only the allosteric binding site but also the active site. Agents that inhibit the enzyme produce an induced fit, makes the active site unrecognizable for the substrate. Okay, so this is an example of allosteric inhibition, and that's how this glucose 1-phosphate and AMP, they internally control the regulation of the conversion of the glycogen to the glucose 1-phosphate subunit. Okay. Now I will go to the next problem. So problem number 12 says, which statement is false regarding the induced fit hypothesis? Your options are: number A, substrate specificity range is high. For the in case number B, the active site is rigid. Number C, more accepted model for the enzyme substrate complex than lock and key. And number D, conformational change occurs in the enzyme and the substrate. Which of the following statement is false regarding the induced fit hypothesis? Earlier, we have discussed about this induced fit hypothesis now and then. So I think all of you can answer this question very easily. I will wait for some time, then check the chat box. Let me know A, B, or C, or D. Which of the following statement is false regarding the induced fit hypothesis? Number A, substrate specificity range is high. Number B, active site is rigid. Number C, more accepted model for the enzyme substrate complex than the lock and key. Number D, conformational change occurs in the enzyme and the substrate. Which statement is false regarding the induced fit hypothesis? Okay, let me check the chat box. I think all of you can answer this question easily. Number B, number B, number B, number D. Okay, so I think number B is the most given answer. Okay, only given answer for this question. So let me check if it is correct or not. So the correct answer will be yes, it is number B. So active site is rigid. This statement is false regarding the induced fit hypothesis. So basically, induced fit hypothesis is another model for the enzyme substrate complex formation. So in the presence of the substrate, the active site undergoes a change which in its conformation to allow the better fit between the active site and the substrate. So this is, let's say this is the enzyme and this is the active site of the enzyme and this is the substrate. So whenever the substrate is closer to the enzyme, what it will do? The enzyme will change its conformation in such a way that now it can accommodate the substrate very perfectly, and then this means the enzymes are highly specific for this reaction they catalyze. Okay, and after the binding, it will just catalyze the reaction, and then product are formed. So the substrate specificity for this induced field hypothesis is very high. It is the correct answer. Active site is rigid. It is not correct because induced field hypothesis we think or we take the active site as not as a rigid but the flexible one. More accepted model. It is also correct. Then conformational change occurs in the enzyme and substrate. It is also correct. Okay. So yeah, statement number B was the false one. So problem number 30, it says that ankymeric assistant is commonly known as number A, neighboring group elimination, number B, electrophilic substitution, number C, addition elimination, and number D, neighboring group participation. So ankymeric assistant is commonly known as what? Your options are number A, neighboring group elimination, number B, electrophilic substitution, number C, addition elimination, and number D, neighboring group participation. Which of the following is the correct answer? Ankymeric assistant is commonly known as what? So ankymeric assistant is commonly known as what? Let me check the chat box. Okay, number D is the only given answer. Let me check if it is the correct or not. So ankymeric assistant is, yes, number D, neighboring group participation. So neighboring groups can accelerate the substitution reactions. So now compare the rates of the following reaction. Each substitution is leaving group is OTS or chlorine, chloride by solvent and known as the solvolysis. Okay. So this particular reacts with the water 1600 times faster than this one. This reacts with the trifluoroacetic acid with 3000 times faster than this one. This reacts with the acetic acid 10 to the power 11 times faster than this one. And this reacts with the acetic acid 670 times faster than this one. So nearby groups can evidently increase the rate of the substitution reaction significantly. Now you may be thinking that we are okay. Let us just skip it out. We have pointed out that catalyne stabilizing group in the center makes SN1 reaction very fast. For example, this one reacts with the nucleophile 10 to the power 6 times faster than this one. And this so in the 4 examples above, it is not the reaction center itself functional group change, but the carbon next to the reaction center. We call this group as neighboring group. The mechanism by which they sped up is known as the neighboring group participation. And okay, so how this neighboring group participation happens basically? So SN1 reaction with the oxalimethyl chloride or this one this is the different group so this example is not the neighboring group participation reaction. Okay, this is the cationic intermediate formation, ease of the cationic return formation because of this reaction is faster, but this is the neighboring group participation. So how it will happen? There is a sulfur has a this has a lone pair which will attack this position, and then the chloride can leave easily, and there is a 3-member ring intermediate and form, and then product formation is faster. But in this case there is no sulfur, so no lone pair of the sulfur which can assist the elimination of chloride from the system. So in both cases ionization of the starting material is assisted by the lone pair of an electro-rich functional group. In the ether in the first example is assisted by formation of the pi bond. However, in the second case the neighboring group participation which is forming the cyclic intermediate and accelerating the reaction. Okay. So ankymeric assistant is the process by which neighboring functional group assists the expansion of the leaving group by the intermediate covalent bond formation. For like this one, if the sulfur atom was a part of an active site of the nucleophile such as methionine, the CCl bond were a part of a substrate, then the RO formation of the and the RO minus were the HMO generated by the enzyme catalyzed deprotonation of water. This would represent a covalent catalysis of the enzyme catalysis reaction, and the covalent adduct represented by the episulfonia chromium intermediate. Okay, so this similar kind of anchimeric assistant is shown where in several enzyme substrate complex where the reaction mechanism follows this kind of mechanism. Okay, now I will go to the next problem. So problem number 14 says Lineweaver-Bark plot is associated with which one? Number A, straight line with the slope of minus km by Vmax. Number B, parabola with the intercept of 1 by Vmax. Number C, straight line with the slope of km by Vmax. And number D, parabola with the intercept of minus 1 by Vmax. So Lineweaver-Bark plot is associated with which one of the following? Earlier, we have seen the Michaelis-Menten curve or Michaelis-Menten plot of the enzyme catalyzed reaction. So there are several other enzyme catalyzed reaction plots also. Like Lineweaver-Bark plot is the second most important. There are several others also, which we will discuss in the next class. So for now, let me know Lineweaver-Bark plot is associated with which one? Number A, straight line with the slope of minus km by Vmax. Number B, parabola with the intercept of 1 by Vmax. Number C, straight line with the slope of km by Vmax. And number D, parabola with the intercept of minus 1 by Vmax. Lineweaver-Bark plot is associated with which one? Let me check the chat box now. Number A, number C. So I have two different options again, number C and number A. Let me check which of the following is the correct answer. So Lineweaver-Bark plot is associated with number C. Okay, straight line with a slope of km by Vmax. So the problem related to the Michaelis-Menten kinetics is that fact that there may not be sufficient data points to determine whether the curve of the Michaelis-Menten plot has reached to a maximum value or not. This means the values for the maximum rate and km are likely to be inaccurate. More accurate values of these properties can be obtained by plotting the reciprocals of the rate and the substrate concentration to give a Lineweaver-Bark plot. So we have known what is the Michaelis-Menten equation. This is the Michaelis-Menten equation we all know is the let me just erase it once. Yeah. So in case of the Michaelis-Menten equation, we know it is the V0 equal to Vmax or Vm. I am writing it. Okay, Vmax. Let me write it. V0 is the rate of the reaction equal to Vmax, which is the highest rate, into the concentration of S, so the substrate concentration divided by Km plus substrate concentration. So this is the Michaelis-Menten equation we have already know. So Lineweaver-Bark plot is nothing but the plot of the 1 by rate or 1 by V0. So if we just take the reciprocal of both sides, so we will get like this. We will get Km by Vmax, and we will get here 1 by Vmax. Okay, we will get Km by Vmax. One position we will have Km by Vmax, and or another position, and okay, sorry, here we will also have a 1 by substrate. Okay, and then we will have plus 1 by Vmax. Okay, so if we compare it with the y equal to mx plus c, y equal to mx plus c, so we will see that when plotting the 1 by V0 with 1 by S, we will get this thing where the slope m is the Km by Vmax and the intercept is the 1 by Vmax or 1 by rate max. So this is known as the Lineweaver-Bark plot or the double reciprocal plot. Okay, nothing. Just we have to just take the reciprocal of the both sides of the Michaelis-Menten equation to achieve this Lineweaver-Bark plot. Okay, so next class we will do this mathematical equation questions with this Lineweaver-Bark plot. So now The next problem number 15 says which of the following statements is false? Number A, both receptors and enzymes have active site. Number B, only enzymes have active site, not the receptors. Number C, after binding, the reaction takes place in the enzymes. Number D, after binding, no reaction takes place in the receptor. Which of the following statement is false? Which of the following statement is false? Number A, both receptors and enzymes have active site. Number B, only enzymes have active site, not the receptors. Number C, after binding, the reaction takes place in the receptor. Number D, after binding, the no reaction takes place in the receptors. Okay. You have to identify the statement which is false. Let me check the chat box now. Okay, number C, number D. So I think I have got multiple options as answer for this question. I can see number D, number A, and number C. Three different options I have. I can see. Okay, let me check which of the following statement is false regarding the receptors and enzymes. So the false statement is number B. Okay, only enzymes have active site, not the receptor. This statement is not correct. Why? Let me show you. So a receptor is a protein molecule usually found embedded within the plasma membrane surface of the cell that receives chemical signal from the outside of the cell, and when such chemical signal bind to a receptor, they cause some form of cellular or tissue response. Okay. So here we have the cellular, it is the receptor ligand. This is the receptor which is present on the outside of the cell, and there are signal transaction via the secondary messengers and ligand which are the primary messengers also. And this receptor ligand interaction, they induces the cellular responses for the various by the various mechanisms. So most of the receptors are the membrane bound proteins that contain an external binding site. Now, for hormones and neurotransmitters, binding results in the induced fit changes of the receptor conformation. This triggers a series of events, ultimately results in the change of the cellular chemistry. Neurotransmitters and hormones do not undergo the reaction when they bind to the receptors. They depart after binding unchanged once they have passed their message. The interaction that bind the chemical messenger binding site must be strong enough to allow the chemical message to be received and weak enough to allow the messenger to depart. Binding groups are the functional groups present on the messenger molecule which are used for binding it to the receptor binding site. Binding regions are the regions of the receptors binding site which contain functional groups capable of forming the intramolecular bonds with the binding site or group of the molecules. So both the receptors and enzyme have the active site, it is correct. Only enzymes have active site, it is not correct. Okay, because the active sites are present both in the enzyme and the receptor. But after binding, reaction takes place in the enzyme but not in the receptors. Okay, although the receptors are active site and the messenger molecules ligands, they bind to the active site of the receptor, but no reaction takes place. It only triggers the events or the passes some kind of chemical message through the receptors. Okay, that is why option number B is the incorrect. Now problem number 16, it says which of the following agent does not have any effect of its own. Your options are number A agonist, number B antagonist, number C partial agonist and number D inverse agonist. So which of the following agent does not have any effects of its own? Agonist, antagonist, partial agonist, inverse agonist. Which of the following agent does not have any effects of its own? Agonist, antagonist, partial agonist, inverse agonist. Let me check the chat box. I think I will have some answers there. Number B, number B, number B. So I have got all the answers as the number B. Okay. Let me check if number B is the correct answer or not. So the agent which does not have any effects of its own is, yes, it is number B, antagonist. So basically, all the receptors or different receptors is defined as a macromolecule or binding site located on the surface or inside the effector cell that serves to recognize the signal molecule or drug and initiate the response to it, but itself it has no longer function. The following terms are used to describe the drug receptor interaction. Agonist, an agent which activates the receptor to produce an effect similar to that of the physiological signal molecule. Inverse agonist, an agent which activates the receptor to produce an effect to the opposite to the direction of the agonist. Antagonist, an agent which prevents the action of the agonist on the receptor or the subsequent response but does not have any effects of its own. Partial agonist, an agent which activates the receptor to produce submaximal effect but antagonizes the action of the full agonist. And ligand, any molecule which attached selectively to particular receptors or site. The term only indicates affinity or ability to bind without the regard of the functional change. Agonist and competitive antagonist both are ligands of the same receptor. Okay. So number B was the correct answer. Now problem number 17 says which of the following enzyme catalyzes the biosynthesis of acetylcholine? Your options are number A, acetylcholine esterase, number B, acetylcholine synthase, number C, choline acyl esterase, and number D, choline acyl transferase. Which of the following enzyme catalyzes the biosynthesis of acetylcholine? Number A, acetylcholine esterase, number B, acetylcholine synthase, number C, choline acyl esterase, and number D, choline acyl transferase. Which of the following enzymes catalyzes the biosynthesis of acetylcholine? Okay, let me check the chat box now. Number D, number D, number D. So in maximum cases, I have got the number D as the answer. Okay, let me check if number D is the correct answer or not. So the enzyme that catalyzes the biosynthesis of acetylcholine is very good. It is number D, choline acyl transferase. So basically, the cholinergic signaling system. So first, let us check what is happening here. Okay, so this is basically so it shows this. So this particularly what happens shows what happens in the synapses involving the acetylcholine as the neurotransmitter. Okay, so this figure, it is showing that the synapse between two neurons, this one, and the events involved in the message as a transfer from one neuron to another. Some general processes takes place when the message is passed from one neuron from a neuron to a muscle cell. So the first stage involved in the biosynthesis of acetylcholine, which is this one. Okay, acetylcholine is synthesized from the choline and acetyl-CoA. So this is the choline and this is the acetyl-CoA. And in the end of the presynaptic neuron. Okay, so in the end of the presynaptic neuron, the acetylcholine is synthesized by the choline and the acetyl-CoA. Okay, and then what happens? The reaction is catalyzed by the enzyme known as the choline acyl transferase. So this is the structure of the acetylcholine. Then acetylcholine is incorporated into the membrane-bound vesicle, which is means for the specific transport of the protein. And the arrival of the nerve signal leads to the opening of the calcium ion channel, that an increase of the intercellular calcium concentration. This induces the vesicle to fuse with the cell membrane and release the the transmitter into the synaptic gap. Acetylcholine crosses the synaptic gap. This is the synaptic gap which is crossed by the acetylcholine, and then binds with the cholinergic receptor, as resulting the stimulation of the second neuron. Acetylcholine moves to the enzyme called acetylcholinesterase, which situated on the postsynaptic neuron. This is the postsynaptic neuron, and which catalyzes the hydrolysis of the acetylcholine to produce choline and acetic acid. Choline takes up by the presynaptic neuron by the transfer of protein to continue the cycle. So cholinergic nervous system involves the nerves which use the neurotransmitter acetylcholine as the chemical messenger. This include the motor nerves to invert the skeletal muscle nerves to synapse with the other nerves in the peripheral nervous system or PNS, and the parasympathetic nervous system inverting the cardiac and the smooth muscle. The two types of cholinergic receptor are there: muscarinic receptor present in the smooth and cardiac muscle, and nicotinic receptors present in the skeletal muscle and the synapse between the neuron. Acetylcholine is hydrolyzed by the enzyme acetylcholinesterase, which deposits the cholinergic receptor. The hydrolytic product choline is taken up by the presynaptic neuron and acetylated back to acetylcholine. This cholinergic receptor and the enzyme acetylcholinesterase are useful drug targets. Acetylcholine cannot be used as a drug because it rapidly hydrolyzes to the acetylcholine enzymes. By the acetylcholine, it shows no selectivity for the different types and subtypes of the cholinergic receptors. Okay. Now I will go to the next problem. Problem number 18 says, which of the following is not an example of the G protein coupled receptor? Your options are: number A, muscarinic receptor; number B, adrenergic receptor; number C, kinase gated receptor; number D, opioid receptor. Which of the following is not an example of the G protein coupled receptors? Number A, muscarinic receptor; number D, adrenergic receptor; number C, kinase gated receptor; number D, opioid receptor. I will give you some time. I will give you a minute. Let me know in the chat box A, B, C or D which of the following is the correct answer. Which of the following is not an example of G protein coupled receptor? Let me check the chat box. We don't have much time, so I'm just doing it a little quickly. Okay, I have got few answers. Number C is the answer given by most of you. So now let me check if number C is the correct answer or not. So there is the following receptor which is not an example of G protein coupled receptor is, yes, it is number C, kinase gated receptor is not an example of G protein coupled receptors. So the G protein coupled receptors are some of the most important drug targets in the medicinal chemistry. Indeed, some 30% of all drugs in the market act by binding to these receptors. In general, they are activated by the hormones and slow acting neurotransmitters. They include the muscarinic receptors, adrenergic receptor, and opioid receptors. The response from the activated G protein coupled receptors is measured in seconds. The slower than the response of the ion channel, but faster than the response of the kinase gate linked receptors, which takes the matter of minutes. These are the large number of different G protein coupled receptors interacting with the important neurotransmitters such as acetylcholine, dopamine, histamine, serotonin, glutamate, and noradrenaline. Other G protein coupled receptors are activated by the peptide and protein hormones such as the enkephalins and endorphins. The G protein coupled receptors are membrane bound protein that are responsible for activating the proteins called G protein. So this is the cell membrane. The G protein coupled receptors are embedded in the cell membrane and they are activated by a protein which is known as the G protein. The later proteins act as a signal protein because they are capable of deactivating the membrane bound enzymes. Consequently, activation of a receptor by a chemical messenger influences the reaction take place within the cell. The receptor protein is embedded with the membrane with the binding site for the chemical messenger exposed by the outer surface. On the inner surface, there are another binding site which is normally closed. Now, when the chemical messenger binds to its binding site, the receptor protein changes shape, opening up the binding site in the inner surface. This new binding site is recognized by the G protein, which then binds. The G protein is attached to the inner surface of the cell membrane and is made up of the 3 protein subunits. But once it binds with the receptor, the complex is destabilized and the fragments to a monomer and a dimer. This then interact with the membrane-bound enzyme to continue the signal transduction process. There are several different G proteins which are recognized by the different types of receptors. Some of the activated by subunits by the G protein have an inhibitory effect on the membrane-bound enzyme, while others have a stimulatory effect. Nevertheless, the mechanism by which the G protein is activated is fragmentation is the same. There are substantial amplification of the signal in the process, and activated receptor activates several G proteins. Okay. So remember, the kinase gated receptor is not a G protein coupled receptors, and these G protein coupled receptors are very, very important receptors in the case of the medicinal chemistry also. Okay. Now I will quickly go to the next problem. Problem number 19 says ATP is required as a cofactor to provide necessary phosphate group, and active site remains long open for the messenger molecule is bound to dash receptor. Fill in the blanks. Again, I will repeat. ATP is required as a cofactor to provide the necessary phosphate group, and the active site remains remains okay. There are two types of remains long open as long as the messenger molecule is bound to the dash receptor. Fill in the blanks. Number A, G protein receptor. Number B, ion channel receptor. Number C, kinase link receptor, and number D, ligand mediated receptor. For which type of receptor ATP is required as a cofactor? G protein receptor, ion channel receptor, kinase link receptor, or ligand mediated receptor? So ATP is required as a cofactor to provide the necessary phosphate group. For which kind of receptor? G protein receptor, ion channel receptor, kinase link receptor, or ligand mediated receptor? I will give you another minute. Okay, since we have not much time remained in our class, let me check the chat box quickly so that I can cover all the things. Okay. Number C is the only given answer. Let me check if it is the correct one or not. So the correct answer is yes, it is number C. Kinase linked receptor is the receptor where ATP is required as a cofactor. So basically, kinase, in the case of the kinase the linked receptor, the receptors tyrosine kinase proteins are there, which is the inactivated monomers. Then signal molecules come, and there is a signal binding site here, and this is the alpha helix in the present in the membrane. This is the first step, okay. And the second step is the formation of the dimer. When the signal molecules are there, the dimer formation happens from the monomer, okay. Then what happened? The ATP is the ATP comes there, and 6 ATPs are required, and ATPs are converted to ADP, and by transferring the phosphate group to the receptor. So activated tyrosine kinase regions are here, the unphosphorylated dimers, and fully activated receptor tyrosine kinase. This is the activated phosphorylated dimer. So now this activated relay proteins are there, which induce the cellular responses. Okay. So in case of this kinase gated receptor, first there is this monomer are there. Then after binding the signal molecule, dimers are formed with the monomer, and this particular dimers are activated by the ATP, which transfer the phosphate to this particular tyrosine residues in the kinase gated receptors, and this induces the cellular responses. Okay. Now, last question of today's session, which says that cholinergic receptors are mainly of dash types. You have to fill in the blank. Your options are number A 3, number B 2, number C 7, and number D 4. So, cholinergic receptors are mainly of how many types? 3, 2, 7, or 4? So, cholinergic receptors are mainly of how many types? 3, 2, 7, or 4? Let me check the chat box. I think I will get some answers in the chat box. Okay, so I have number B as the only answer. Let me check if number B is the correct one. So the correct answer is yes, it is 2. So cholinergic receptors are mainly of two types. So this is the classification of the cholinergic receptors. So mainly cholinergic receptors are two types: ligand-gated ion receptors and G-protein coupled receptors. Okay. So G-protein coupled receptors, for example of the ligand-gated ion channels are the nicotinic acetylcholine receptors, which are of the two types: are central nervous system and automatic ganglia in the peripheral nerve, which is the neuronal nicotinic acetylcholine receptors, and muscle nicotinic acetylcholinesterase receptors. The neuronal ones can be a homopentameric or heteropentameric, and these are all the subunits of the particular receptors. Okay, the classifications are important. If you can remember, if you can memorize it is fine. If otherwise it is also fine if you cannot memorize all these things. But remember these things like for the muscarinic receptors, these are of 5 types: M1, M2, M3, M4, and M5, and all these classifications. So there are different subunits, all these things. But I don't think deeper and remembering the most all the things are more important. I don't think so. But you have to know how the receptors work, what are the particular signal molecules or activators of the each and every receptors. These are the things are the important. Okay. So this was the last question of today's session. So what did we learn today? Today we discussed about the enzyme catalysis. We understood the basics of the enzyme catalysis. Then we discussed, we did some enzyme kinetics, some mathematical questions also. Then we discussed about the receptors. Okay. So today it was our week 2 live session. So now I will share my email ID with you here in the chat box. So So this is my email ID. Any problem, any doubt, you can let me know through email also, or there is a discussion forum in the NPSWM web portal also. You can find the you can post your queries there. So where to find this class recording and PPT? So if you go to the here in the announcement section where the left panel is there, there will be a problem solving session recording panel. From there, if you click there, you will directly redirect to a page where you will find this particular class presentation which I have presented today, and along with the video of today's class. Okay, all these things are posted there, so you can go through the materials. So today it is our week 2 live session. Every Friday we will have this kind of live session from 4 p.m. to 6 p.m. Okay, and next week we will meet at the same time and we will discuss the week 3 course content. Till then, go through the week 2 course material and also the materials which I have provided today, and let me know if there is any problem regarding these things, and by this email also. So I will close the session for today. If there is any query, I can take one or two. If not, I will close the session for today, and note my email ID. You can just let me know through the email your any doubt, any query. So if it is fine, I will close the session for today. Thank you all for joining. Thank you all for participating in the question answer session also. Like this, if every Friday we will do this kind of session, and I think it will be beneficial for you. This kind of question, if you solve it by yourself, you will easily answer the assignments and also the final questions. Okay. So thank you all. Let me know if there is any problem regarding this class or anything by the email. Now let's meet on the next Friday from this time. So I will close the session for today. Thank you so much.